So I am presenting on behalf of the PICAL Consortium, and specifically I want to mention Kavita Reddy from the Africa Health Research Institute, who performed all the laboratory work. These are my conflicts of interest. Especially of interest is that this research was funded by VIV. Well, we all know that the reservoir in infancy is influenced by some factors, as mentioned today, as age initiation of ART, time on suppressive ART, and time when suppression is achieved. What we are adding in this talk is a year-to-year description of the reservoir up to 4 years, factors associated with specific time points, and factors associated with dynamics, and the fact that we found around 7% of what we call excellent controllers, which mean a combination of sustained viral load suppression plus good CD4 for a long time, for at least 12 months, and plus undetectable reservoir. The EARTH cohort, this comes from early antiretroviral treatment in HIV-prenatal infected children, was a cohort with children from South Africa, Mozambique, and Mali, 220 patients. These children were enrolled in the first 6 months of life, and all of them were treated less than 90 days after diagnosis. They were monitored 2, 6, 12 and 24 weeks after enrollment, and biannually up to 4 years. The age of ART initiation was a median of 34 days, with a median follow-up time of 44 months. We performed the reservoir in total PBMCs through digital droplet PCR, and PCR amplification in a minimum of 5 million PBMCs once a year. And the proviral HIV DNA was undetectable if it was below 1 copy per million PBMCs. We defined sustained viral load suppression as viral load undetectable in the last 12 months, at least with 3 different measurements, and immunological control as no significant immunosuppression during the last 12 months, again in 3 different measurements. We did a regression analysis to analyze the factors associated with lower HIV DNA at different time points, adjusted by baseline HIV DNA, so therefore you're not going to see that baseline HIV DNA is a factor associated with DNA in the long run, and mixed effect Poisson regression modeling to analyze the HIV DNA dynamics. So this is the cohort. We could analyze 207 patients, and we could analyze those who had more than 12 months of follow-up, 155. Unfortunately, there was a 12% mortality, and there was also a significant dropout of 1/3 of participants. And as you can see, if I can, I'm not sure if I can, well, it doesn't work, but yeah, looks like it works now, yeah. Here, you can see that 1/3 of participants had sustained RNA viral load control, and in the right part of the slide, you can see that 12% had undetectable last DNA. This was 19 participants, and in the middle, 40% had immunological control. We combined undetectable DNA and sustained RNA control, and this happened in 15 participants. And if we combine sustained RNA control plus undetectable DNA plus immunological control, we found 11 participants, which is 7% of the cohort. This is the evolution of the reservoir. you can see a steeper decline in the first year, and then a decline which is not so steep. And as you can see here, the slope is declining, a negative slope in 73% of the participants, and especially in blue, those who achieved sustained virological control had a steeper slope than those who didn't achieve virological control. Now I'm going to present the factors associated with a lower reservoir at different time points, and then with the dynamics. The model included baseline, as you can see, different features, and especially time to suppression. We cannot see it very well, but and mother social issues, and time to suppression. This is 1 year after enrollment. 2 years after enrollment, the main factor was baseline viral load. And 3 years after enrollment, again, the main factors were baseline viral load and the first year slope, but also mother social issues, and if the participant was a female. And this is a specific time points. And if we look in the dynamics of the model, we see that some of these factors doesn't influence so much, but are taken over by serial viral load over time since ART initiation, and again, mother health issues. So why do we find different factors in the specific time points they differ from the dynamics? We think that the interpretation may be that the dynamic factors, as viral load over time or mother issues, seem to modify the reservoir evolution and reduce the influence of baseline factors. Also, of course, this may be related with different sample size, as you can see, and it may be also biased by death and loss to follow-up and sample availability. These were the evolution of the 11 excellent controllers. In red, you can see the evolution of the reservoir, and in blue, the evolution of the viral load, and in yellow, the evolution of the CD4. All had PMT-CT, started ART at a median of 29 days. They had variable adherence. As you can see, most reported good adherence, but some of them reported 67%, 37% of visits. They don't report it an optimal adherence. From the baseline perspective, they all had lower baseline viral load, higher CD4 counts, and lower DNA. And in the follow-up, in general, they had better numbers relating peak viral load, CD4 nadir, better DNA slope, and faster time to viral suppression. So in conclusion, early virological parameters as pre-ART viral load and initial slope of DNA are important predictors of medium-term viral reservoir size in early treated children with HIV. Predictors may vary depending on the time of measurement. Dynamic factors as viral load over time and environmental factors as region or mother issues seem to modify reservoir evolution and reduce the influence of baseline factors. Sex biological and social differences should be explored to understand mechanisms of HIV reservoir control. And around 10% of children who start ART within 6 months of life can achieve undetectable reservoir. Not only those who are extremely early treated, as in Impact 1115 or in the Tatello studies that treated or Uquaningo cohort that started treatment really early. We can see that even we start a little bit later, which is the standard of care, these participants can actually achieve also a negative reservoir or undetectable reservoir. We believe that these participants represent ideal candidates for analytical treatment interruption trials targeting cure. We think that a specific joint strategy for curing children is needed. We would like to thank families, researchers, and funders as always. Thank you very much.